Well, thank you very much. It's a really fun day here for me to be in this multidisciplinary Nanotechnology Research Institute. So hopefully there's something for everybody here as we go through. And I'm standing up here as one person, but of course all the work I'm going to present was really done by my research group. So of course I want to acknowledge them, right up front. If you have any questions, please, I guess it's okay in the filming process here to stop me as we go along and ask me at the end. But if you still have questions afterwards, please feel free to email me right there. So everything we do in the lab kind of revolves around gold. And it's an interesting story how it all started. But you can actually go back thousands of years and realize that people thousands of years ago knew that finely divided metals had different optical properties than the bulk metal. So a good example is this cup here. This is a Lycurgus cup. It's a Roman 4th century AD cup. There's metal work here, but then there's glass inside, and the glass has this rosy color. Okay, and I am wearing a different shirt. Okay, good. So this rosy color, it turns out, is from gold particles that were finely divided. So the Romans knew this. You could take gold and beat it with a hammer until it's micrometers thick. It still looks like gold, and you disperse it into molten stained glass, and molten glass, and then it adopts this beautiful color, which was called red gold at the time as a pigment. So they knew even back then that this was the case. And then fast forward to 1857, and you can go to London, and in the Royal Institution in London, there's a basement full of memorabilia from people who used to work there, and one of the people that used to work there is Michael Faraday. And so this is his chemical bench, and this solution right here, I think it's ether is the liquid, and he's the first person we know of to take a metal salt and reduce it to the metal using phosphorus as the reducing agent. And so this is this nice rosy color again. And these are, again, from very small gold particles. Okay, so his solutions have been sitting there since 1857, and they're still pretty stable. So you wouldn't want to be the graduate student on that project, right, to see how stable particles are over a long time. Okay, so that's a long time that people have known these things. And it turns out now we have a pretty good understanding of why this is. What's the reason for these properties? And so again, you can go back, and now over 100 years only, to Gustav Mie. And so what did Gustav Mie do? He said, I have Maxwell's equations, which were known at that time, and I can solve Maxwell's equations for a specific case. So Maxwell's equations tell us light is an electromagnetic wave. And you can analytically, without a computer, solve them for the case of the small conducting sphere. So small to him, smaller than the wavelengths of visible light, which are hundreds of nanometers, conducting meaning metal, sphere meaning sphere. So all you need to know to solve this equation, these equations are the radius of the sphere, dielectric constant of the metal, refractive index of the medium. And you can solve it. And if you put in gold and, 20 nanometers and glass or ether, you will get the rosy red color, okay? So it's only been in the last 20 years that people have done the same kind of calculation, and you do need a computer for this, for different shapes of particles that are not the simple conducting sphere. And you can get quantum mechanical about this sort of stuff too if you want to go deeper. So in other words, we don't want to be much bigger than a few hundred nanometers. If you're bigger than a few hundred nanometers, then you're just going to look like a bunch of gold flakes in solution. But you don't want to be too small either. So we learned over time that if you're too small, your particles are not going to have what we call now a plasmon. So how do we picture this? Here's my small conducting sphere. Here's the electric field vector of light coming in. And we have these huge oscillations of these conduction band electrons, okay, in the metal. And that's responsible for the strong absorption, and it turns out also scattering features that we get from these metal particles. So any element in the periodic table will do this, okay, under the right conditions. But if you want to be in the visible part of the spectrum, you're mostly going to be copper, silver, gold, which are all in the same column in the periodic table. And copper oxidizes and silver photooxidizes, so we like gold. So if you're too small, though, you're going to lose it. You're not going to have enough conduction band electrons to support this, and your particle is going to look more and more like a molecular cluster and have kind of discrete states. So anyway, for us, then we're always going to be bigger than about 4 nanometers and smaller than a few hundred nanometers. Okay, so that's the size range of interest. So in my group, probably what we're most known for is making a bunch of these differently shaped metal particles. And so these are rods. And so this is where you do need a computer to solve Maxwell's equations. So here we are making almost spheres. And you see it's that same rosy red color we had for Faraday and for this Roman cup that I showed you. And these are about 20 nanometers in size. And then as we go larger along the axis here, we are increasing the aspect ratio of these particles. So we're making them longer and longer in one direction, and the width isn't changing too much. So that's the aspect ratio here is length divided by width. So increasing the aspect ratio, it's still a pretty tight range if you want to think about it that way. And these solutions here are 1 nanomolar solutions in water of our particles. So this tells you these are strong absorbers, right, compared to a lot of molecules. Extinction coefficients 10 to the 9th per molar per centimeter on a per particle basis. And if you take spectra, here they are. So here's our 1 plasmon band we had before for a sphere, from Gustav Mie, from Michael Faraday, et cetera. And then as we collect spectra for these guys here, you see we have 2 bands now. And how people understand this, our 1 plasmon band is splitting into 2. So we're absorbing light and scattering light, it turns out, too, along the short axis and the long axis of these particles. And so as you make the particles have a higher aspect ratio, the longitudinal plasmon mode, the long axis mode, shifts to lower and lower energy or longer wavelength. So the shape of the particle dictates where the light's absorbed. So that's going to be an important feature that we'll see. So this little picture here looks like a galaxy. If I told you it was a galaxy, you'd probably believe me, but what it actually is, it's a dark field optical micrograph of our nanorods immobilized in a polymer gel. So all we're doing in this experiment is we're sending white light into the gel, and we're just collecting light that's scattered out to the side. So it's just Rayleigh elastic light scattering, why is a sky blue kind of scattering. So the field of view is about 3 millimeters, but all little stars in the sky, those are the particles. So that's not an absorption and a re-emission of light, it's just a scattering event. So there's no bleaching, there's no blinking, and you can follow them around even in a turbid matrix like this polymer gel, which gives rise to this kind of misty colors we see here. So some people are motivated by fundamentals. I mean, it's actually pretty interesting to think about how do these little crystals grow and how do you control them and all that stuff. And some people are motivated by applications. I get the sense this is more of an application crowd here. Maybe I'm wrong. But just to let you know, what are the applications? And we'll be seeing some of this later. It all depends on the light absorption properties of particles. So here's my rod. I come in with light that is resonant with one of those 2 plasmon frequencies. And what do I get? I get these electrons moving around, which means I get a big electric field near the surface, which means if you want to do molecular photophysics as a function of electric field, you have the perfect system. So the most famous example of this is surface-enhanced Raman spectroscopy, where a molecule whose Raman modes aren't very visible, okay, when you have a monolayer, suddenly light up when you sprinkle particles on top because you're coupling the plasmons from this thin metal film to these particles here. I'll show you this later. Some photophysical properties of molecules, if you're too close to a metal surface, it's not going to be good, so you want to be a certain distance away before you start seeing something. So then you have to build up spacer layer chemistry here. So up here what we're going to do is make bad fluorophores good ones by another phenomenon called surface-enhanced fluorescence. So that's using the electric field that you generate here. But it turns out if you come in with a lot of light, like I said, these particles absorb a whole lot of light. What do they do with all that energy? They don't emit light. They dissipate it as heat to the environment. And then you can do things with this heat. So again, I'll show you later. We can do things like get molecules to pop off. So this is sort of a controlled delivery kind of deal. Or if you want to go all the way to a very medical application, you see this poor mouse here with this tumor, and you can squirt particles on there and shine laser light where they absorb, and you will ablate the tumor thermally. So nothing's going to be resistant to chemotherapy in that kind of scenario. So there's a lot of interesting activity up here, a lot of clinical trials going on, and so we don't get to this level, but we're very interested in the heat aspect in terms of what you can do in terms of getting phase transitions to occur that are interesting. Okay. So it turns out we did develop some patent stuff on making our particles, and so now you can talk to me. I'll send them for free, okay? Or you can talk to Aldrich and buy them. I didn't actually look up these prices until recently. So 1 mil of solution for a standard kind of solution like I showed you, they charge $1,000. So my students, when we do things on the 50 mil scale, it's $50,000 worth of stuff, and now they're afraid to do anything. Okay, so I'll show you details of the synthesis in a little bit, but let me just say on the bio side, we have all these cool applications, right? There's various modalities where you can do chemical sensing based on plasmon band position, which does shift as the local refractive index of the medium changes. If you aggregate the particles, there's big shifts. Or you can do this electric field and do stuff with molecules. Tracking, you can imagine from the little starry night picture. Drug delivery sounds a little weird, okay, for a hunk of metal to deliver drugs, but because of this photothermal thing, people are more interested. Okay, so there's a lot of activity in this kind of space. And then we have issues. And what are the issues? And this is where we spend a lot of time. Okay, you generate heat. How much heat would that be? You generate fields, like how strong are they? How far out do they go? Et cetera. And if you're going to do things that are going to involve some kind of sensing, then you are probably going to want to do something like this, attach some kind of molecule to your particle that will then transduce a response in some way. Okay, but as I'll show you, how you choose to do that actually affects the ability of your molecule to do its job on the surface. And then if you're going to go to the cellular level, you got to worry about things like toxicity, and then things get overcoating your particles after you've made them. If you do things with live cells, they go in, how long are they there, what do they do? And then we're finding over here some kind of weird stuff, I'll show you at the end, about how the presence of nanoparticles in the environment around cells affects their behavior. It doesn't kill them, it just changes what they do. And if we can understand that, that could be super cool, right? But it all starts with these particles. So let me just give you a very brief summary of, 10 years of work on how to make these particles well. So we developed a process a long time ago called seed-mediated growth. So what's the idea? So as you know, anybody making colloidal particles from molecular precursors in solution, you have to worry about nucleation and growth and all that stuff. And nucleation is hard. So what do? We skip it. So what we're going to do is take a metal salt. I'm showing it for gold, but you could do other metals here. So we take our metal salt, and we're going to be in water, in air at room temperature. We're going to have some ionic things around here to stabilize the particles when they grow so they don't agglomerate, but it could be lots of things. And then we're going to come in with a strong reducing agent, sodium borohydride. And the idea is that you're going to quantitatively convert all your gold-3 ions into gold-0 atoms that are going to form these little seeds, these little tiny particles. And indeed, that's what happens in about 30 minutes, and you get these sort of solutions that are kind of reddish-brown, and they don't actually show a nice plasmon band because these particles are too small to show a plasmon. Okay. So then we take them out, put them in a fresh pot with more metal salt, okay, and now a weak reducing agent, ascorbic acid coming up here. And the idea is that ascorbic acid at room temperature doesn't have the thermodynamic juice to reduce gold-3 to gold-0. So we don't want to make new seeds. We want to grow on the existing seeds, and the redox potential of a surface-bound species is different from the free solution. So we will autocatalytically grow on the seeds, is the idea. Now, if you do what I just said with nothing else, you're just going to get bigger and bigger spheres at some level. So what we need is something else in the pot to direct the growth, and that's what this surfactant stuff is here. It's CTAB, so cetyl trimethylammonium bromide. Cetyl's an old name for a 16-carbon chain. There's the trimethyls. This thing makes rod-like micelles in water by itself, which was our original idea, which turns out to be wrong, that it would be like a soft template. We have our soft rod-shaped thing in solution. Our seeds assemble in some electrostatic fashion, and then we pack together a polycrystalline rod. Now that's not what happens, but that's why we picked this stuff, and it actually does produce rods. Anyway, so in early days we did the following. Here's my metal salt with this CTAB stuff. I add my weak reducing agent first, so because it turns out order of addition matters here, which is telling you things are very kinetically controlled. And then I add my seeds last. You go from gold-3 to gold-1, there's a color change there. You add your seeds, and now in this pot, a whole bunch of colors start changing, which is telling you you're growing in like 30 seconds. Okay, things are growing pretty fast. You have multiple growth pots sped up, and so you go from 1 aliquot to here, 1 aliquot out, 30 seconds here, etc. And if you go all the way for gold, you can make these nice long rods, which are about 600 nanometers long and 20 nanometers wide, which is sort of like 2 tobacco mosaic viruses end to end. Now, this sounds great, right? And you can stop it along the way and get smaller aspect ratio materials. Silver, you can do this, it takes off and makes really long wires, but you kind of lose control over the length. This sounds great. What's the problem? The problem is yield. Yield is terrible. It turns out most particles do not elongate. Only a small proportion do, but of the proportion that do, they seem to keep going. Okay, so then, around the same time as other groups, we found that if you add an additive metal ion impurity into the solution, that controls the growth. What's the additive metal impurity? Well, here I'm showing silver nitrate, but it's the same idea. Here's my growth solution. Okay, I have this metal salt, this CTAB stuff. I come in with my weak reducing agent and this other stuff, silver nitrate in this case, and then I add my seeds, and then in one shot, I get a whole bunch of rods. That was a heavily purified micrograph. This is a completely unpurified micrograph. So your spherical side issue is gone, okay, but now you have a whole bunch of rods, which is good, but now you can't really grow them any longer. You have to go back to the beginning, add a different amount of silver. Okay, so as you can imagine, we spend a lot of time doing all kinds of chemistry, in here, which I will skip for the most part. So I'll just tell you, though, that even though we've learned how to do this pretty well for a while now, we keep improving things. So here's a slightly different prep. If you want details, quantities, here we go. So here's making the seeds. Okay, you can change the pH. That does change things, etc. And then now, what's the difference? The big difference is the weak reducing agent, not ascorbic acid, it's hydroquinone. It has a different redox potential, but kind of so what. And our tuning of the aspect ratio comes from tuning in this range of silver nitrate concentrations. Okay, so you do this, but you get gorgeous particles, okay? And now we have high yields of plasmon bands pushing out to 1200 nanometers. So 1500 nanometers of telecommunications wavelengths. Our old prep with no additional metal ion, actually you could get out there, but it's kind of iffy. And now we have much better control. And the particles are just absolutely gorgeous when you do TEMs of them. I mean, it just warms your heart, okay, to see these gorgeous particles, right? So, I mean, they're just beautiful. Okay. Okay, so I will mention very briefly that we're trying to analyze things. And one of the things that nobody likes to talk about in this kind of world of colloidal and particle synthesis is variability, batch-to-batch variability, irreproducibility. If you've ever tried to prep from the literature, except for ours, you know, you have problems, right? So here's an example. So I just mentioned that adding more silver, you get a higher aspect ratio, longer longitudinal plasmon wavelength. So here is one experienced person in my lab. Here's the silver. They go along. It looks all nice and linear. See, great. That's all nice. Then if I take everybody in my lab for the last 5 years and put them all on one graph, here's what I get. Wavelength, silver. Okay, any one person here is their own little line. But if you were a new student in the lab and you're trying to make rods that absorb at 785 nanometers, a common laser wavelength, you've got a factor of 5 variability in this reagent compared to everything else being the same. So you wonder, why is this? And what can we do about it? Okay, so then I encourage you to read this paper where we do kind of a design of experiment thing. And the idea here is that instead of varying one thing at a time, that's what we normally do, right? Vary one thing at a time, one thing at a time, one thing at a time. When you do that, you may not realize it. You're assuming nothing else is coupled to that system. But if you have synergistic effects between things in your pot, guess what? You're going to miss things. So anyway, here in this paper, we do 2 things at a time, maybe 3 things at a time. And we actually try to kind of learn who needs to be near who to get this growth to happen. So anyway, you can ask me later about details there. Anyway, no matter which way you do it, in the end, the particles come out and they're in water, and there's a ton of this surfactant around, and they have, it turns out, a ton of this stuff on their surface in the form of a bilayer. Lots of experiments here. What's really happening at the base gets interesting. It's a chemisorb bromide, we would say. Okay, and then the big fat cationic head of the surfactant has an electrostatic association there, and we're in water. We got hydrophobic tails, and we got a ton of the surfactant, so we make a bilayer. So in the end, we have a high positive charge, net positive charge on these particles. I'm kind of showing the ends look like they have less of that stuff. Indeed, there is some indirect evidence in the literature that this could be the case. Again, you can ask me later because I don't think I'll have time to talk about it. But once you know this stuff is here, then you got to think, oh, okay, then I should realize it's there. What can I do with it? So you can stuff small hydrophobic molecules in there, right? You can try to replace this, cover it up, build off of it, et cetera. So there's a really long article here about the surface chemistry once you know it's there. Okay, so this is all by way of introduction to the particles. So now I've got 4 little short stories here, something for everybody. Okay, so the first one is physics. Well, I'm not a physicist, so this is as close to physics as I get. So Michelle, you can, hammer on me if it's not physicsy enough for you. So here we're thinking about the electric fields. So here's the surface of my particle, here's distance from the particle, and let's say I shine light at the right wavelength, I generate electric field. How far out does it go? Okay, everybody would say, if you're 150 nanometers away from the surface, you're done, right? You're not going to feel anything if you wanted to feel an electric field. And then as you get closer, it's going to go up at some particular rate. So let's say we have a fluorophore, okay? And a quote everybody knows, if you take a fluorophore and put it next to a metal surface, its fluorescence will be quenched. Okay, energy transfer, electron transfer, lots of reasons. You can argue about the reasons, but everybody would say you got to be some distance away before the fluorophore will be emissive. Okay, and that's actually the basis for a lot of sensing modalities. In the case of a fluorophore that's actually some known distance away, okay, people are interested in this phenomenon called surface-enhanced fluorescence, which is to say a bad fluorophore, kind of a low quantum yield, becomes a much better one by virtue of the electric field effect, it would be feeling for both the absorption and emission process. So what do people find in the literature from lots of different systems? They find something like this. Somewhere between 10 and 20 nanometers out, you're going to get an apparent maximum. So really this kind of green curve here is the superposition of quenching when you're too close and enhancement when you're some distance away, and then the apparent maximum is somewhere here, 10 and 20 nanometers. How much of a boost do you get when you do this? This is where it gets interesting. You read the literature, factor of 2, factor of 10, factor of 100, factor of 1,000,000, okay? And there's data for all that. So then you wonder which one is, right. Okay, so we'll get back to that in a second. But this is just an example of the kind of thinking you have to do when you're going to think about, photophysical enhancement of some process in a molecule that's going to experience an electric field. Then you got to think about building layers. If you want to get out 10 or 20 nanometers, what do you do, to do that? So one of our favorite techniques for building layers is piezoelectrolyte layer by layer wrapping. So this works great on flat surfaces, and it works well in colloidal particles too. The problem is you have to separate the polymer from the colloidal particle when you're done. So here's my particles that I start with. I can buy this polymer, sodium polyacrylate, PAA. It's got a net negative charge at the right pH. And you can incubate these two together, wait a while, and spin out the particles, resuspend them. They were cationic. Now they're anionic. You come in with a positively charged polymer like this one down here, pH. You wait a while with the right ionic strength and et cetera, right molecular weight polymers here. And then you can separate the particles, and then you go cationic. So you can flip the surface charge between layers here pretty significantly. And on a flat surface, like a microscope slide, you could do this 100 times. Back and forth, back and forth. On a colloidal particle where you got to separate free polymer from the particle, you can maybe get about 10 layers before you start losing too much sample. So how thick are these layers? On a flat surface, by ellipsometry, those particular polymers I just showed you are about 1.5 nanometers each. So that's pretty nice and tight. On our surfaces, when we do this, this is a dynamic light scattering experiment. It's probably easiest to look at this table down here. So without any polyelectrolytes, CTAB all by itself, a molecular model would say 3 nanometers thick is that hydrophobic region. The experiment here says 2.9. By the way, this experiment is coming out of light scattering. It's a little bit of an interesting experiment. Dynamic light scattering, if you've ever done it, the number you get out for the size assumes everything is a sphere, which we know we don't have here. But if you know you have an anisotropic shape by TEM, you can go ahead and get the coating thickness. There's a little formula Luis Liz-Marzan developed, the coating thickness of your layer using this simple experiment. So that's pretty handy. So anyway, we do this, and it's interesting. It's so we have nanoscale control of the thickness here, but you notice it's thin, it's fat, it's thin, it's fat, it's thin, it's fat. So we do know distance, but it's not interesting. And what we think this is telling us is that this is how hydrated these layers are when they go down, which otherwise would be hard to measure in water. Okay, so now we're ready to do some cool experiments. So what's this? This is a molecule here. It's AF something, something, something. This is from the Air Force. This is a nonlinear optical molecule. It is a 2-photon absorber. Why does the military care about 2-photon absorbers? I am sworn to secrecy since I'm in a different country. Okay, I can't tell you why they care. But what they do want to know is what is the ability of this molecule to do its 2-photon absorption process as a function of electric field. So here's what we do. We take our particles. So what is 2-photon absorption, I should say. So here's my ground state. Here's my excited state. Normally you'd say I need at least this much energy to do an optical electronic transition in my molecule. 2-photon absorption, as the name implies, you send in 2 simultaneous half-energy photons to do that process. So you need femtosecond lasers to do this. Anyway, so this molecule does its 2-photon thing at 785 nanometers. And so we're going to make rods that absorb at 785 nanometers. And go ahead and put layers on here and put our molecule, which is the little green things here, further and further away from the surface. So we want to see what the electric field effect is going to be here, right? And there's theories in the literature we can compare to. So we put our molecules on there, okay, and then we monitor the surface change as we do all this wrapping and all that stuff, okay? That's all good. After we make all these molecules, we destroy them, all these systems, and destroy them to quantify how many molecules we have per particle, which turned out to be about 1,000 in this case, except for this one. This is the average of 6 different batches. And this one's always low for some reason we're not sure about, but once you know it's low, you can correct for it. Okay, so now we do the big experiment in collaboration with the Air Force, where we measure the 2-photon absorption cross-section for those number of chromophores on the particle divided by what it would be off the particle. And that black is the data here. So I should mention the organic chemists that make those organic chromophores, they will spend a lot of time doing substituent stuff and maybe get a factor of 2. But you see here, we got a factor of 40 by just putting on a particle, okay? And the data tells you closer is better. If you calculate the electric field of the 4th power, which is what ought to be happening in this 2-photon thing, the red is, there's 2 different curves here. One, if you include this closest data point or not, we're using a program called COMSOL here. So it's pretty clear that, and we're doing the measurement in an interesting way. You can ask me about that later too. Anyway, it's pretty clear that electric field governs all, right? I mean, the closer is better for this particular phenomenon. So if we could stuff that molecule right on the surface or chemically attach it, which we actually spent a lot of time trying to do, then it would be even better if all you cared about was a 2-photon absorption cross-section. But what's kind of interesting is that it doesn't, in a way, make sense when you think about the electric field, like where they are. So this is a calculation of the fields. And so this is 8 layers out, which is as far out as we went. So clearly you don't need to go 8 layers out. And clearly all the action when you shine light at 785 nanometers for rods that absorb at 785 nanometers, all the action's on the end. So what you really want to do is get all your molecules on the end. Now that is hard, okay? And so this is something that we're working in the group on a lot now. And again, we've got some recent results, which I don't have time to show you, where how can you functionalize a rod-shaped object just on the ends and not on the sides? I mean, there's data in the literature that says you can kind of do it. We have the reproducibility issue there too, okay? But anyway, it's a very interesting field to go into in terms of colloidal science. Okay, so I mentioned polyelectrolyte spacer layers. They were great for a lot of things, and especially if you want thin layers. But there's other places where you want to be a little bit fatter. So this is the example of the fluorophore. So now we got a bad fluorophore. It's actually, there's another whole piece out here which I'm not showing called IR dye. So this thing absorbs way out there, 800 nanometers-ish, and it emits pretty close to that. And its quantum yield is pretty low. So here's the experiment. We're going to take our rods and put on now a silica shell because we can make that significantly thicker, and I'll show you how we do that in a second. And then we're going to couple on a silane azide, which we had to make ourselves. Well, actually, we had an organic student make it for us because this is not commercially available. And then we can buy commercially this dye where you can do a click reaction and go ahead and put this whole thing on there. Okay, so then the question is, where does the plasmons have to be and how thick? So remember I said 10 or 20 nanometers, somewhere in there. So here's the whole dye, here's this whole thing. Here's the place where we're going to click. So thank God you could buy this thing, right? So here's the absorption, here's the emission. What we're going to do is make a series of rods where our plasmons are going to be on the blue side, on top of it on the red side of the absorption and emission here. The plasmon peaks are broad, so we're going to be on top of both absorption and emission, which is actually probably a good thing anyway for this. And then we're going to move the molecule further and further out by the silica spacer layer. Okay, so the particles, we know how to do that. And then the spacer layer silica, now this was another place where things that look like they work in the literature actually are a little tough. Even we have a paper long ago of putting silica on gold nanoparticles, although of just only one thickness. The problem here is that these are mesoporous silica shells, and the C-tab actually that's on the particles is actually going to be the template where we're going to hydrolyze and condense silica around that on the particle, which is a little tricky. And it turns out you really need to know your C-tab concentration to do this well because the outer leaflet is on, off, there's dynamic equilibrium. How do you know that? Bromide ion selective electrode is the answer. And once you know where you are there, now you're very reproducible and making really gorgeous silica shells here that are 6, 12, 15, 17, nanometers thick, over a certain range. Okay, so that all worked great. And then we go ahead and put on this dye. So we got 5 different kinds of aspect ratios, 5 different thicknesses. So it's 25 experiments here. And I just want to point your attention to the hot spot. Okay, where do we get the most light out of this dye, if that's all we cared about? Well, we want to be where the plasmons are right on top of the absorption and emission. You want to be right on top. That makes some sense. And then where do you want to be? You want to be about 17 nanometers out. And how much more light do you get out compared to the free dye at that same concentration, all that stuff? It's about a factor of 9. Okay, so now we know. When this is just steady state data here, you always want to make sure you want to do time resolved work to confirm your conclusions here. And this is a lot more tricky than I thought because I thought we'd be able to say a lot of words about the radiated rate and non-radiated rate of the dye pretty easily. But it turns out, right around the same time we were writing up this paper, we found a quantum mechanical calculation in the literature, and the paper was called approximately Calculation of the fluorescence quantum yield of molecules adsorbed onto gold nanorods. Okay, what are the odds? So what they found in their paper, they had these beautiful maps of Kr and KNR varying as a function of distance. So it's really a complicated system in the end, much more than I thought. But what do they conclude? Generally, 15 nanometers out is what they calculate is where you want to be if all you care about is getting more light out of your fluorophore. We find 17 experimentally, so we declare victory. Okay. So this is all by way of the electric fields, that we're using, the particles that we generate when we shine light. You can also use the heat. So here's the experiment. So here's my rods again with that C-tab bilayer again. We're going to do polyelectrolyte layer by layer deposition again. We electrostatically associate another molecule. We had the 2 photon absorber before. This is rhodamine 6G, good fluorophore, good Raman scatterer too. But for the purposes of this little experiment here, the interesting thing about it is every time it binds to the surface, it's going to see this layer of PAA, polyacrylic acid. So this is net positive, PAA is net negative. So if you take PAA and rhodamine 6G by themselves and do a calorimetry experiment, you find their association is exothermic, which should say, according to Le Chatelier's principle, if I add heat to the system, they should want to decomplex. So therefore, first hypothesis, I shine light, I generate heat, the molecule is going to want to pop off. Second hypothesis, if I put more and more layers on top of it, it's going to have a harder time coming off. So indeed, that's what happens. So this is measuring the release. So this looks like real-time data, but it's actually very laborious. We made 785 nanometer absorbing rods. We're shining light kind of like this laser pointer, so nothing fancy at all. And one tube, and we have a dark control tube. And this is measuring the free dye and the supernatant. So one layer on top, 2 layers on top, 3 layers on top, as you shine wimpy light for some number of minutes here. Now, if you're a physicist, you might think this is the most boring experiment ever. Why don't you just put your thing in a water bath? Okay, all right, so you can do that. And our laser is like a 36°C water bath then. But what you have here is control over what wavelengths you use. So this is where we were, rods, PAA, rhodamine 6G. Okay, we had about 1,700 molecules on there. We popped off about 100. Okay, and then as you put more and more layers on top, less and less comes off. But here's the same surface chemistry, but with spheres of gold that do not absorb at 785 nanometers. So that's the control experiment. You shine light on them, nothing pops off because they don't absorb the light. So that's how you have control. So I found out recently that Seagate, the disk drive maker, is using this principle in one of their processes now. So they call it HAMR, heat-assisted magnetic recording. What do? They have all these little bits that have spins, right? That's how you store and read information. And you want to do a phase transition between them to, read your bits. And so what do? They put gold nanorods that absorb it in the near IR. I think it's 1064 nanometers. And you raster with a laser, and you generate enough heat to flip the spins in your bits. Okay, and then voila, we have terabyte storage coming 2018 based on this process. Okay, all right, that's enough physics. Okay, so now we'll go to some chemistry. Okay, so we care about chemistry. We do a lot of surface chemistry on these particles. And so here's a general principle I want to share with you, which is how molecules are attached to the particle surface affects their activity. Okay, a lot of times you wish this was not true, but actually it turns out to be the case. So what are we going to do here? This is an old experiment now, but it's still a good one. So we take an enzyme, trypsin. We're going to attach it 3 different ways to the particle surface, quantify how many we have per particle, and then see how active it is, okay, and using its favorite substrate, which is casein, okay? So that's what we're going to do. So, okay, this is looking pretty familiar. We make the particles, there's that surfactant bilayer. We're going to do 1 round of this polytrolite layer by layer coating. This is an interesting polymer, though. You can buy this one, polystyrene sulfonate maleic acid copolymer. So this is going to give us net negative charges all over the place, which is fine. But it turns out when we do these surface chemistry reactions, you don't ever want to go through the point of 0 charge. We're always in water. If you go through the point of 0 charge, you are likely to aggregate out and precipitate, and we never want to do that. So what we have here is this copolymer. We're going to use the acid groups later, but these sulfonates are always going to give us negative charge, so we always have some charge, so we don't precipitate. Okay, so trypsin is a small protein. I forget if it's got 7 or 11 amines on the surface, but it's net positively charged at pH 7. So the simplest thing to do by far is mix your net negative particles with your net positive protein, and that's what a lot of people do. Right there, antibody, aptamer, whatever they're doing. Okay, fine. Or you can make covalent bonds. So we can make amide bonds between the 7 or 11 amines. I'm only showing one here, but of course there's a bunch. To your acid groups. That's like sophomore organic chemistry, no problem. Or we can do that click thing again, where we have an azide and an alkyne. So here you're going to make amide bonds between the 7 or 11 amines on the surface of trypsin with pentinoic acid. So then you have triple bonds facing the solvent, carbon-carbon triple bonds. That doesn't affect the protein activity at all when you do that. And then we're going to come in here and put on some azide, and you can buy this thing. So this is an amine peg azide. So this is just a little peg unit, 3 units long. Okay, and then we put it on. All right, we do all that stuff, and we quantify everything. And this way was a lot more work, right, this whole click thing. But it actually is kind of worth it. So here's the 3 different ways. How many proteins do we have on there? So these are little proteins in big rods. So about 15,000 proteins would be a monolayer. So we're about halfway to 2/3 of the way filled. And then how active is this protein? We have no control over orientation here. And so you lose a lot of activity. okay, in these two methods, but this click thing is actually kind of worth it. And this is corrected for how much was there. And so then you wonder, why would this be? And it turns out we still don't have a clear answer, because it seems like there's probably several things going on. What are the several things? If I go back, first of all, this little linker here is giving us more space between where the protein is and this hard surface. So you could say, well, the protein's got more room for the substrate to come in, so that's like a steric argument. Okay, and there's evidence for that. But the other thing you could say is, well, the nature of this linker, PEG, polyethylene glycol, everybody knows proteins don't like to be adsorbed to pegylated surfaces, but also everybody knows, why is that? Because PEG is really well hydrated. And if PEG is really well hydrated and you're a protein and you find yourself near a pegylated surface, you are more likely to adopt your native conformation. Okay, so then the argument would be that this system gives us protein that's the right shape, and these two are denatured. Okay, and there is not overwhelming evidence for that either, but there's a little bit. Okay, so that's the point I'll return to later, is that how can you measure well the conformation of your protein on a particle surface? It turns out there are some problems, okay, when you try to do this. Well, another thing that we've been looking at lately is lipids. Okay, so lipids, we know what they are, and what we're going to do is try to make our particles look like little lipid vesicles. Okay, so here's the idea. I'm just going to use spheres here. So I've got my gold spheres. I will go ahead and again, I will choose two different ways to attach my lipids. I'll use electrostatics or hydrophobic van der Waals interactions. So I can come in with my cationic polymer. Now I got a positively charged surface, and I can come in with this lipid or that lipid. Okay, one lipid, another lipid, or the interesting thing, a mixture of the two lipids, okay? And then I've got a little lipid-coated particle. Or I can go ahead and take these particles, add a long-chain thiol, so now I have a very hydrophobic surface, and I can come in with one lipid, the other lipid, or a 50-50 mixture of the two of them. And what's keeping the lipid on here is just van der Waals interactions between the hydrophobic tails of the lipid and the hydrophobic tails of this thiol I put on. People tell me that have done this with other systems, that this one is seems to be more stable in cells, okay, if you're wondering. Okay, but I'm going to do this. I'm going to put these lipids on there, and the question is, for this mixed lipid case, if I put in a one-to-one mixture, do I get one-to-one when I'm on the surface? And how are they arranged? Are they a random alloy of lipids, or do they form domains? Ooh, that's going to be a harder one, okay, to measure. Okay, so we quantify everything, and so we do little particles and bigger particles here. These are just spheres, and it makes a difference which way you did it. So if I use van der Waals in my favor, I put in one-to-one. No way do I get one-to-one. I'm way enriched in this lipid. Same is true here. And in the smaller particles, I don't even quite have enough to go all the way around to make a nice bilayer. If I use electrostatics in my favor, I put in one-to-one, I get one-to-one, but now I actually have a lot of lipids, so I actually have multi-layer adsorption onto this particle. Okay, so these are pretty different looking objects depending on which way you decided to do it. And then the question is, where are the lipids? Okay, can we do like an AFM experiment looking for, okay, these two lipids are the same height, so that's not going to work. We tried a lot of things that didn't work. So here's the thing that worked the best. Here's what it is. So one of those lipids has a carbon-carbon double bond. And people that do a lot of electron microscopy know that carbon-carbon double bonds will react with osmium tetroxide and known electron microscopy stains, so you can visualize them because osmium is a heavy element. So this is the correct lipid. I hope you can see this in the projector here. It's a pretty good projector. So you see the layer, this is the right lipid all the way around, right? So you can get the thickness, you see the striations, it's all very nice, okay, for these different particles here. That looks all good. And then here's the big experiment. This is the correct lipid. This is the other lipid, so you should be able to see anything. So you got to make sure you focus right. And then here's the big result. See that blob on one side? Okay, that's our big evidence that we have a domain of that lipid on this size particle. So we do a lot of statistics, okay, lots of statistics. And what do we conclude? So you can actually get the domain size of those lipids, okay, on the bigger particles, but for 20 nanometer particles, we never saw these patches. So either the technique's not good enough to resolve it, or, and more interestingly, right, the curvature is too high to support a patch, right? So then we have a random array of these lipids. So again, the object would look different to like a cell, okay? Okay, so we think about lipids, we think about proteins, and then we think about something like this. So this is a very interesting protein, alpha-synuclein. It's one of the most abundant proteins in the human brain, and it has no known function. Isn't that interesting? But people do know, if you have Parkinson's disease and you look on the brains of people with Parkinson's disease, you find these fibrils, okay, which are made of this protein, alpha-synuclein. It's thought part of its action is binding to lipid vesicles, okay, which is why we connect lipid vesicles here. But it's also biophysically a very interesting protein. By itself in solution, it's a random coil, doesn't have much of a structure. But if you incubate it with 25 nanometer phospholipid vesicles, it goes alpha helical, as judged by circular dichroism. And if you incubate it with small primary means like this one, it goes beta sheet, as judged by circular dichroism. So two very different conformations. Well, the minute we hear any molecule is going to change its shape when it binds to some kind of nanoscale thing, you know, we are there. So what we're going to do is do some experiments with this protein. So here is our gold sphere. Now we're just going to use spheres again. Okay, I'm going to have a very innocuous anionic surface here. No lipids, just something really boring. Okay, then I come in with my protein, and I'm basically here, I'm going to just do a titration. Okay, what is the binding constant of this protein? How many proteins are here? Stuff like this. Okay, lots of ways to do that. You can look at the plasmon band shifting, dynamic light scattering, and then if there's multi-layers of protein, you can come in with high salt and then go around the loop again. So there's an overall protein corona, people would say, and then there's a hard inner corona that's not exchangeable, and then a soft outer corona that is exchangeable, which might be there, so we got to check all that. And then at this stage, then you say, all right, what's the conformation here? Let's do that circular dichroism experiment, alpha helix, beta sheet. Turns out that's a hard experiment because these particles absorb a lot of light. This is one problem with these particles. That experiment is hard. It turns out we found a way to get around it later, but at the time, what we had to do was do something else. So we came in with our friend trypsin here who will digest things. And what we're going to do is chew the solvent-accessible pieces of this protein and do a mass spectrometry like footprinting experiment to see what is solvent-accessible. Okay, so we do that. So everything's looking good. The protein's well behaved. Plasmon bands are shifting. Dynamic light scattering is going. Okay, we get numbers for 20 nanometer and 90 nanometer spheres again. So this protein binds pretty tight, okay, overall. And then the inner part is like 100 times as tight. For the smaller particle, there's about 420 proteins on average for each particle, and most of them are stuck pretty tight. You go to a bigger particle, everything's bigger. There's like a big web of proteins, though, associated, and only about 1/10 of them are stuck pretty tight. Okay, so that's just kind of good to know where you are. And then the question is about which way are things pointing, and what kind of orientation and conformation do we have here. And we do this whole mass spec thing. Okay, it's complicated. We're looking for holes in the pattern and so forth. But in the end, what's the bottom line? We do have a preference here for these initially anionic particles. The N-terminus of this protein is protected pretty uniformly from all of our chewing, and therefore the C-terminus is facing out. And if you look at the sequence of the protein, what do you notice? Well, the reds are the positive charges. So then you think, well, I just wasted a year of my life doing this experiment that I could have predicted from first principles based on electrostatics. Okay. All right, well, if that's true, we can do everything all over again and wrap our particle up with a cationic surface, you know, and just go, right? And then we can predict what'll happen. It turns out this is a disaster, okay? It's a disaster of epic proportions. You're titrating, you're not reaching equilibrium, you're going to bimodal huge distributions here, so what's going on? Now you can do CD in the solution because not just the protein bound to the surface, but all the protein in solution is going beta sheet. So this is what's happening. We're seeding the formation of this protein. We're making these huge flocks, which actually can just see come out of solution, that look red, which tells you the particles are still well separated and all. Anyway, this seems bad. And if you remember, we had primary amines promoting beta sheet conformation of this protein. And here this particle had all kinds of primary amines, and it's doing the same kind of thing in a concentrated manner. So this would maybe suggest that maybe all cationic particles are bad in terms of biology, which actually is a conclusion some people come to. But wait, okay, we have a new hope, okay? So I got two particles again. Here's a negative one, this is just citrate. And here, now this is an interesting molecule, only made for the first time in 2012. It's a long-chain thiol, and now it's got a quaternary ammonium. So this particle will have the same positive charge as that other polymer wrap thing, but it's a very different kind of surface. Quaternary ammonium is not primary amines. It's got a lot of hydrophobic character buried down in here. And now we're getting more serious now about these surface chemistry changes. We do our titrations on this guy, and this is looking good. So this cationic particle is well behaved. The protein binds, and it tops out and all that stuff. You can get a binding constant, et cetera, all very nice. And now we're getting more serious. So now we're doing some NMR with my colleague Chad Rienstra, who, guess what, does solid-state NMR of alpha-synuclein fibrils, okay? So he has N15 labeled protein, so we're able to do this two-dimensional experiment here. So this is with the anionic particle. So blue is the free protein and gold is with these anionic particles. I have to say, this sounds like trivial. I mean, if you're a chemist, people do NMR all the time. They do NMR all the time on millimolar amounts of molecules in solution. We have nanomolar amounts of particles, and we want to see what's on the surface, which is maybe micromolar if we're lucky, and NMR is not sensitive. So heroic experiments on the part of my student Wayne Lin figured out how to concentrate our particles by a factor of 300 in solution without them aggregating, and the magic buffer here is heaps, in case you want to know, and that we don't want to aggregate the protein either, okay? Anyway, what do we see? It's interesting. I mean, I don't do a lot of NMR, so I'm thinking, oh, we'll get the structure at atomic resolution of the protein on the surface. No, that's too much to hope for at this stage. What you do see is residues that are close to the particle surface are broadened so you don't see them, so you can only see stuff far away. And what do we see? The N-terminus is broadened so you don't see it, and the C-terminus is you can see. So the structure of the protein isn't that different, actually. You're just losing the N-terminus with anionic particles, just like we saw before with that mass spec technique. And then if we do the same thing with these new cationic particles, it's beautiful. It flips. So now we see the N-terminus, and now we don't see the C-terminus. And so molecular dynamic simulations also help confirm this picture, that we're able to show which way this thing is pointing, okay, on the particle. And so what we're doing right now, and we haven't finished this study yet, my student made 10, 20, 30, 40, 50, 60 nanometer particles with lipid molecules, 10, 20, 30, 40, 60, mutant proteins, okay, it's this huge matrix of experiments, which is turning out to be really interesting, because alpha-synuclein deforms lipid vesicles when it binds, and you can't deform these little guys, though, so what will it do? Okay, it's an interesting story, coming soon. Okay, but that leads me into biology. Okay, so we're going to think about the cell here. And the cell, we know cells, we're made of cells, they're alive, they do things, and if you have a nanoscale object, okay, depends on the size of the object, there's many different processes that are pretty well known to the cell biologists about how things get into cells. There's all kinds of different penocytosis, macrocytosis, endocytosis, there are all these names, some require energy, some have ATP, I mean, a lot of stuff is known. I'll just mention, if you want to get things into cells and you care about clearance, meaning stuff coming out and not getting stuck in the liver or kidneys, just to let you know, if you were interested in this, you want to be smaller than about 10 nanometers to clear the kidneys in a human. Okay, if you're bigger than that, you're going to get stuck there. Maybe that's not a problem, but probably it'll be a problem. Okay, so we do stuff with cells and particles, and one of the experiments you can do with this is this one, which is a pretty fun one. So here's what's going to happen. I'm going to show you a movie. So I've got a little dish. I've got a substrate on the dish. It's collagen. Okay, so it's a protein that cells like to bind to. And these are boring cells, human dermal fibroblasts. And they have proteins that recognize collagen, and they will crawl around. And we got particles everywhere here, vanilla-flavored particles, nothing fancy. And this is dark field microscopy. So the cells aren't labeled with anything. But what's going to happen in this movie is the cells are going to get brighter and brighter and brighter as they suck up these particles. And you'll see dark trails left behind where the particles were. Okay, that's what's going to happen. Let's see if this works. So it's working. All right, so we're sped up by about a factor of 8 from real time here. Okay, and we're not doing anything to these cells. We're just letting them do what they want to do. Okay, they're crawling around. No problem. You know, they're moving around. They're stretching out their, whatever, their feet and arms. Okay, you see this big hole, right? That's the nucleus. It's pretty big, isn't it? So if you want to believe these two-dimensional images of three-dimensional objects, you could say, okay, the particles aren't getting in the nucleus. But we didn't design them to do that. And you can see the dark trails left behind where the particles were. So you look at these movies, and then you wonder, well, why would they want to do this? I mean, if I was a cell, I would just want to sweep this stuff out of the way. But no, maybe they're like a baby, and everything goes in the mouth, okay, doing this. But you can make a lot of measurements at this stage. You could do things like scrape the cells off, see how much gold they've acquired by ICP mass spectrometry. It's a lot. It's like 500,000 particles per cell, which is a whole lot of mass to be dragging around. The other thing you can do is measure individual cell trajectories. So that's actually pretty interesting. The most interesting thing, so a couple different cells here. So PC3 is a prostate cancer cell line. HDF is human dermal fibroblast. So this is mean square displacement over time. So this is black is no particles. These are all different sorts of particles at the same concentration, different surface chemistries, different shapes, et cetera. Anything you feed these cells slows them down, which makes sense because they're taking up all this extra mass. But if you go over here to these, quote, normal cells, some particles make them slow down, and some make them speed up. Now that's pretty interesting, even though they're taking up all this extra mass. Okay, so you talk to cell biologists, and then you want to say, the cells aren't dying. In fact, they're zipping around a little faster than they were before. So then you're forced to do things like this, okay, do some microarray gene expression PCR stuff, and here we've got a nice suite of particles with lipids and without and so forth. And then you're forced to do things like this and look at the changes in gene expression of 14,000 genes as a function of particle concentration. Okay, for 2 different cell types. Okay, the only thing to get out of this picture at this stage is 2 things. First of all, you can't predict what's going to happen from one cell type to another, and also, lots of stuff is changing. So red is upregulated and blue is downregulated. Okay, so this sounds really bad, right? I mean, these cells are like turning into mutants or something. You notice control here, control has color. You might wonder, why does control have color? Shouldn't it be white? It turns out this is how, when you present this kind of data to biologists, they always want to see like the raw data. Okay, so yeah. Then if you actually, control for the control, it's not so bad, right? The 14,000 genes. There's only, I don't know, a few hundred that are changing. And every particle is a little bit different, okay? Some they have in common, some they don't have in common for these genes. You might wonder, if you don't do a lot of gene stuff, does the dose affect the number of genes expressed, right? Is there a relationship? Okay, so that's number of particles per cell, and here's number of genes differentially expressed. Well, okay, you could draw a line like that. You could also draw a line like that, or you could, okay, no, there's no correlation, okay, for uptake and gene expression. Okay, now if you're like me, you're wondering what's the mechanism, right? I have a result, which is pretty interesting. What's the mechanism? This is where I don't have a ton of time to tell you the gory details, but let me just tell you 2 ideas that we have. So we know, right, we already know, we can absorb proteins or lipids to particles. Right? We know that'll happen. In the context of the cell, what we're saying is that unbeknownst to us, you're changing the concentration of molecules in the cell, right? Probably dynamically over time as this happens. And the cell will respond to such changes, right? Cells are looking for molecular cues. They're taking in nutrients. They're expelling waste. They're doing a lot of stuff. And so if you are, sort of interfering with that, the cell will respond. And not at the level of killing it, but you've actually done things like sequester nutrients, perhaps, that it was looking for. Or maybe you've sequestered things that made it slow down, and now it's speeding up. Okay, the other thing is that we have hard little objects, and everything that the cell is kind of around is more soft. And so then you think, oh, well, maybe we're changing the nature of the environment the cell is in. And cells do respond to the mechanical nature of their environment. There's a whole field in biology called mechanotransduction. So that's another thing. Okay, we have evidence for both of these things. So I think I'm supposed to end in like 5 minutes. So I'll have to just blow through this part really fast. Okay, but we do have evidence for both of these things. What kind of evidence do we have? Well, if you wanted to say, what can I do to not make stuff stick to a particle, your default response would be, I'm going to put on some PEG, polyethylene glycol ligands. Okay, this is a different kind of experiment. This is with cardiac cells, but we had responses of these cells when we had just our traditional old polyelectrolytes. We go ahead and put on PEG, and the response is mitigated, but not completely zeroed out here in terms of this is a different kind of cell behavior. I didn't show you the details here. And if you do some gene stuff, again, I'm not showing you the original data, we were at factors of 5, and now we're factors much closer to 1. Okay, so you can mitigate but not reduce the response if you make your particle somehow impervious-ish to things sticking to it. Okay, so that's some evidence for that idea. And then the other evidence is this whole mechanical thing. Okay, this is a pretty weird experiment. I'll go through it very quickly. So what we have here is we have cells in a little 3D culture in here, and we put them inside a bigger collagen gel where there's particles or not. And the question is, will the cells move out from the inner gel to the outer gel? Well, actually, they want to do that. They want to get uncrowded. So they will move out. And the question is their migration affected by particles being in the outer gel? Okay, that's the question. Okay, it turns out so then you're counting cells. Okay, it's a pretty boring experiment. And it turns out there is a pretty big difference here in the terms of the cells migrate. So more cells move when the matrix has these particles. And it turns out there's a whole lot of mechanical stuff going on, so then you need to do rheology and microscopy and figure things out. And indeed, the mechanical nature of the matrix is softer with these particles out there. And again, the cells will respond, okay, in ways that are reasonably well understood, but now we're connecting nanoparticles to the mechanical properties there. So I'm going to skip this kind of stuff. The only thing I will say here is the way they move is also different. So moving through the gel normally, the cells are kind of elongated like they should be, going like this. But when there's particles in there, they move in a more rounded fashion. So to the cell biologist, this is a mesenchymal to amoeboid phenotypic transition, which the cells could undergo depending on conditions. And we're saying here, these particles are a condition. So again, for full understanding, we want to actually kind of connect particles to cell behavior. We know cell behavior depends on physical properties of the matrix. Nanoparticles affect that. Okay, so there's this like loop, right, that we have to go around for full understanding. Okay, I have 30 seconds to tell you my last story, but that's okay. It's a short story. Ecology. Okay, so what we're going to do here is ask the question, where do engineered nanomaterials go in the environment? If I have a model ecosystem, where do they go? Can I predict it? What do they do? Okay, but this is just where do they go and how much is there? Okay, so it turns out we're going to collaborate with NOAA here, the National Oceanic and Atmospheric Administration. They have a lab on the coast of Charleston Harbor, South Carolina, where they pump seawater in from the harbor into tanks that have organisms that you tell them you want, and then you can do an exposure of your pollutant. That's what they are mostly looking at. And then you can quantify where the pollutant has gone in your system. Okay, so the estuary is where the river meets the sea, so we have big concentration gradients in salt, salinity, pH, it turns out. If you stand at the estuary and stick a pH meter in the water, your pH will vary from 8.3 to 4.5 with the tides in this place. It's pretty huge. Okay, so we have, okay, so here we go. We got 3 tanks. Okay, it took us a year to reserve these tanks. We get 3 replicate tanks, 366 liters of seawater per tank. We get 1 shot, 1 dose. Okay, what dose are we going to pick? That's a good question. We're going to pick, okay, the question is, all right, here's my particles again. Okay, our particles are like the size of a virus. If a virus got into an ecosystem, what would its concentration be? That's kind of hard to answer, but it turns out it's a few parts per billion. Okay, so that's what we're going to do, a few parts per billion. So we make a boatload of particles for 1,200 liters of seawater. We've got organisms, we've got a tidal cycle, we got clams, we got shrimp, we got all this stuff. Okay, and then we let it go for 2 weeks, our 1 dose. Nothing dies. Okay, that's good, nothing dies. And then we do all this like high school biology dissection experiments to figure out where all the gold went. So this is 8 months of ICP mass spec, published some time ago now. And my biggest fear here, right, in seawater, are just CTAB-coated particles are going to aggregate salt out, go to the bottom, we never find them. Right, in seawater, not true. About 10% stay in the water column, about a quarter go to the sediment. The big sink in the living food web, the microbial biofilms, the slime, that's where a lot of the stuff ends up. And with a little bit going to the clams, you know, which are filter feeders. Now, some people had said that roots of plants would take particles up because they're very small. That would make some sense, but that doesn't seem to be happening here. Okay, and this is just 1 particle, okay? Thank you for that. Yes, I'm almost done. And then we do everything all over again, okay, with data. That's all data point number 1, okay, 3 years of work. Here's data point number 2. This is where we were, okay, it's written a little bit differently. Now we do everything all over again. We do 1 round of our polyelectrolyte coating on the same kind of particles, and we do it all over again. And everything is different, okay? So, you know, the quantities are all different. Plants are coming online. Okay, we got problems with fish hatching. Okay, anyway. But the point is, this is only 2 data points after all this work. So really, I'm still trying to work on NOAA to do 1 more round of like a, I don't know, like a neutral particle. And then we can have 3 points and make a line of something. Okay. But anyway, as far as we know, this is like the first couple of studies where we change the surface chemistry rationally for an engineered nanomaterial ecological exposure. Okay, so what have we learned? What have we learned here? On the physicsy side, okay, we learned the plasmon tunes the molecular photophysics depending on spacer distance from the metal. We also have some photothermal release rates. On the chemistry side, right, we're saying that, you know, your molecules on the surface might not necessarily be wild type when you have them on there, and behooves us to check that. And again, we're getting very excited about NMR, learning about some orientation on curved surfaces. On the bio side, right, we learned that we're changing genes upon exposure to particles, which sounds like a bad thing, but if we understood it better and we had some cell going down a pathway we didn't want it to go down, we could maybe correct it, right? That would be a pretty powerful thing. And even though we don't understand why, so far we can say that initial surface chemistry, before the 2 weeks of exposure in a natural water system, the initial surface chemistry still influences the environmental fate of these particles, even though it's going to be a complex story as to figure out how to predict it. Anyway, well, I want to thank my group for all the people that I've mentioned here. I haven't mentioned all their names in particular who've done all this work, collaborators across all across the U.S. really, and then support from these agencies. But I mostly want to thank you for your attention, and if we have time, I'll be happy to take any questions.
Questions? Yes.
First of all, thank you very much for this very informative lecture on nanoparticles. My research team is working on both nanoparticles, both nanosecurities for drug delivery.
For what? Huh?
For drug delivery.
Yep.
And one of the challenges that we have is, I think you talked about it a couple times, aggregation.
Yep.
Particularly like when we try to concentrate the number of nanoparticles.
Yep.
I was wondering if you faced any challenges like this, and how did you overcome them, or what would you suggest to avoid aggregation of both nanoparticles?
Sure. So there's a couple of strategies. So in the NMR stuff I mentioned, where we had to concentrate our particles a lot. So it turns out the actual, I'm sure somebody knows the deeper reason for this, but operationally what we found is heaps buffer. It seems like you can concentrate things a lot in heaps without them aggregating. Now, why that's the case is not clear to me, but just if you want like a buffer to check out, that would be a good one. The other thing people do here is try to have, of course, non-ionic ligands, right? So you can buy, pegylated ligands that maybe end in a hydroxyl or a methoxy. If you don't care what the surface is, you probably care what the surface is, though, right? So anything you can do to reduce the charge would be beneficial, right? Those are the two general strategies people would have.
Okay, so you're saying like reduce static interactions, or?
Right. So you might wonder, why do particles aggregate, right? Well, if you're putting them in a salty solution for some bio stuff, right, then you're going to salt things out. Like when you purify a protein, right? How do you purify protein? You salt it out. You go through the isoelectric point, and it'll have then no net charge, which means it's going to aggregate in water and then precipitate out. So that's the sort of thing that can happen when you have a highly charged particle. So if you can neutralize charges to some extent, then you could mitigate that. But again, there's a lot of like old school biophysical theory about those sorts of things, right? In the case of our CTAB coated particles, one mechanism by which they aggregate is the outer leaflet is very dynamic. And if it stays off long enough, then you start exposing hydrophobic residues, and then they will interact with other hydrophobic parts, and then you have a sort of a big giant van der Waals thing coming out. And so in our cases, yeah, if you do something like put it in different solvents other than water, you start stripping off the outer leaflet, exposing a very hydrophobic thing, which starts to aggregate, right? So there's different ways things can aggregate. So for your particular system, you have to make sure you understood which one it was.
I was interested in this cell uptake in particles.
Yep.
And they start shining. Can you explain why? What happens with this shining?
Oh, all that's happening is we're concentrating the particles in the cell. So we didn't add anything. So there's particles all over the place, above the cell, around the cell, under the cell, everywhere, and they're not big enough to scatter a whole lot of light on their own. But as the cells suck them up, okay, we get more and more particles in a smaller space, and I don't think they're, well, to know if we're aggregating them or not, we'd have to do some spectroscopy on the scattered light. But even if you didn't aggregate them, you basically just have a higher concentration of them in a small spot, so then they become much more visible, because now all the ones in that focal volume are scattering. So it's just a concentration effect. So you can do the same thing if you added a bunch of salt or something right there, you'd see a bunch of them aggregate.
Maybe one last. Yes.
Very quick. Maybe we'll have a chance to talk more. But in case of your protein it's work on called nanoparticles, and there is a sort of this controversial issue of possibly plasmon enhanced chirality. I'm not sure if you have any evidence of that, if that was sort of focused of your work, and whether you have resonant conditions at all.
Well, so just to tell people what's going on here. So if you have a chiral molecule that maybe gives you a circular dichroism spectrum in the UV, there are people that have evidence that if you have that on a plasmonic particle like gold, suddenly the plasmon band is chiral, right? Like you can see a CD in the visible or near IR from your particle, even though the particle is not chiral, or it didn't used to be anyway. Actually, I got to say, we have tried that. We're like, oh, we have these proteins. Every time we try to see any CD signal on the plasmon, we never see anything at all. And I've talked to a number of people who are like, oh yeah, I don't have any trouble, and we still have not figured out what they're doing so differently from what we're doing. I mean, maybe we're completely incompetent. That's always a possibility, but.
Whether it's just a surface chirality, whether it involves only surface. But I mean, if it's plasmon, that would mean that the entire plasmon is.
Yeah, that's right. That's right. That's right. It's very interesting.
And so do your molecules show an enhancement in CD signal due to the presence of gold?
Yeah, so no, so the for the protein stuff, you got to be in the UV there, and that actually gets a little complicated. Yeah, right. Yeah.
Okay, let's thanks Catherine again. Let's